Today I'll be exploring perplexities in Alzheimer's disease research. So we have all of these jokes and funny stories about how older generations can never remember anything. But say you're with a family member or an older friend, and at some point you realize they're not really functioning the same way that they had been. Maybe they forget where they put their keys every time they set them down. Or maybe they're having trouble with remembering what day it is or what day of the week it is. Or they can't really recall a conversation that they had just a few hours ago, or they have to ask the same question over and over and over again, even within a conversation. Eventually, these memory concerns are brought up to a doctor, and that witness account alongside a PET scan and oftentimes a cognitive test is how Alzheimer's disease is diagnosed. And at the time of diagnosis, Alzheimer's may have been progressing for upwards of 10 years prior to actually seeing symptoms and being diagnosed. And so what exactly is Alzheimer's disease? So Alzheimer's is a progressive neurodegenerative disease where the death of nerve cells and loss of synapses in the brain lead to a loss of cognitive function that is always fatal in the end. And so Alzheimer's affects approximately 6.7 million Americans and about 50 million people worldwide, and accounts for about 60 to 80% of all dementia cases. And dementia is categorized as a complete loss of cognitive function, where people don't even know how to eat or sometimes drink water properly, stuff like that. It's really, really sad. And currently, there are no known cures or no known causes. We don't know the reason why behind neurons die. We just know they do, and when they die, it leads to this cognitive decline. And so to better illustrate the prevalence of Alzheimer's in America, here's a little pie chart of the prevalence of Alzheimer's in senior US adults. So that's anyone over the age of 65, and that is about 12%. And as age increases, the slice of pie of people with Alzheimer's also increases. So at 85 years old, there's about 36 of the population that has Alzheimer's, which is wild. And then these are MRI scans. On the left is a typical brain. And as you can see, it's kind of full. If you can see my mouse, there's not much gaps or space anywhere. On the right is an Alzheimer's disease patient. This is a post-mortem brain. You can see there's just huge gaps everywhere, and there's a lot of extra space. And in this red circle is where Alzheimer's neurodegeneration starts, and this section includes the hippocampus and amygdala, which are parts of the brain that are tied closely to long and short-term memory as well as emotions. So there are really two types of Alzheimer's disease diagnoses or categories. And the first is early onset, which is Alzheimer's diagnosed before 65 years of age. These are typically this kind of Alzheimer's only accounts for about 1% of all Alzheimer's cases, and it's caused by genetic mutations in the amyloid precursor protein, presenilin 1 or presenilin 2. And then the second is late onset, which is any Alzheimer's diagnosed post 65 years old. And there's not really late onset is pretty elusive because we don't have any 100% telltale signs that is exclusive to Alzheimer's or any known root cause. But there are known factors that increase the risk of developing Alzheimer's, and the biggest of these is age, followed closely by the presence of the ApoE4 allele instead of an ApoE3 or E2 allele. And then smaller risk factors include bad cardiovascular or dental health, brain inflammation, and surprisingly, gender. If you're female, you have an 11% chance or higher chance of being diagnosed with Alzheimer's later on in life. And this isn't even an exhaustive list because the body is an interconnected unit, and there's no one isolated system. You change something else is probably going to be changed. You just don't know it all the time. And again, there is no known cure for Alzheimer's. However, there are 5 FDA Approved Therapeutics to slow the symptoms and progression of Alzheimer's. And so there are 2 broad categories. The first is acetylcholinesterase inhibitors, which include donepezil, which was approved in 1996, rivastigmine, approved in 1995, and galantamine, which was approved in 2001. And the other category is NMDA antagonist, memantine, which is used to treat severe Alzheimer's, and that was approved in 2003. And then there's also this combination drug of Namzaric, called Namzaric, which is a combination of donepezil and memantine, and it's used to treat moderate to severe Alzheimer's as well. But hold up. did I really just say the last FDA approved drug for Alzheimer's was in 2003? And the answer is kind of yes, I did. And that's the question we'll be exploring today. Why haven't there been any new therapeutic treatments approved for Alzheimer's since 2003? And we're going to explore this by first looking at what the current therapeutic strategies for treating Alzheimer's is, and then looking at where the current and new future of Alzheimer's disease research is heading, and trying to find like the links between the two, and then maybe where like future insights could go. And so the two main hypotheses used as models for Alzheimer's treatments currently is, well, the cholinergic and then NMDA receptors again. And so first we're going to look at the cholinergic system, which kind of focuses on everything surrounding acetylcholine, which is a vital neurotransmitter involved in attention, motor functions, and memory, and is additionally important for some synapse function. And I mean, there are other things that acetylcholine does, but those are the big ones, especially memory, of our interest. And so an imbalance of acetylcholine is often associated with Parkinson's, myasthenia gravis, and Alzheimer's disease as well. And particularly in an Alzheimer's brain, there's evidence of depletion of cholinergic production, and less acetylcholine production or too much acetylcholine breakdown is often associated with cognitive decline and neurodegeneration. And this is kind of the cholinergic system pathway, and we'll start on top of the image where choline and acetate are turned into acetylcholine by acetylcoenzyme A and choline acetyltransferase. And then this newly made acetylcholine will then bind to an M1 protein, which helps facilitate synapses between neurons. And then excess acetylcholine, the stuff that doesn't bind to M1, is then broken down by acetylcholinesterase back into choline and acetate. And choline isn't really mass produced in the body. We just kind of get it from this like recycling of the system, and also someone's diet can introduce a lot of choline into the system. And so when there's not enough acetylcholine in the system, then there's a loss of synapses because it doesn't bind to as much M1 protein. And therefore, it'll eventually lead to cognitive decline and neurodegeneration. And so donepezil comes in and it binds to the acetylcholinesterase inhibitor and prevents the breakdown of acetylcholine. And so in a depleted cholinergic system, the increase of available acetylcholine means there's more binding to the M1 protein, and therefore an increase in synapse between neurons, which then halts the progression of Alzheimer's symptoms progression for just like a short bit of time. Donepezil looks like this, and so it's composed of these three sections. On the left, there's a benzyl group, so this aromatic green. In the middle, there's this piperidine group with a nitrogen that has all of its valence electrons taken up by bonds. And then on the right, there's this indinone group with O-methyls attached on the very right. And how does donepezil bind to acetylcholinesterase, you ask? Maybe. Well, on the left is an image of acetylcholinesterase, and it's a dimeric protein, meaning there are two identical subunits that kind of just like click together, and that's how it is. And those arrows are pointing to the active sites, which is typically where acetylcholine will bind to. But when donepezil is introduced, donepezil will bind to that active site, and it'll kind of block acetylcholine from going there and being broken down into choline and acetate. And then on the right is just like a zoomed in image of this. And so the oxygen methyl groups are up in like the upper corner of the image, which is a little hard to tell, but they bind through hydrogen bonds to tryptophan 286 here. And in the middle of the molecule, which is around here, which is where that piperidine with that nitrogen is. So that nitrogen will formed water-mediated hydrogen bonds with tyrosine 337 and 341. And then the benzyl group that was all the way on the left in the prior image just kind of takes up extra space to like be extra sure that acetylcholine can't bring in. And so that's kind of how it binds and inhibits acetylcholinesterase. And rivastigmine and galantamine work in similar ways where they bind to this active site and make it so acetylcholine can't be broken down. And what's cool about the similarities between them is they all have like this nitrogen that has all of its valence electrons taken up in bonds and then can be used for hydrogen bonding. And there's also these oxygen molecules on each of the drugs that can also then, oh my god, that can also then interact with the acetylcholinesterase and have hydrogen bonds and water-mediated hydrogen bonds to further block acetylcholine. Cool. Now, the second hypothesis used in current treatments for Alzheimer's includes the N-methyl D-aspartate, or NMDA receptors. And the theory is overexcitation of NMDA receptors promotes neurodegeneration, vaguely. So how does this NMDA receptor work? Well, first, agonist amino acids, glutamate and glycine, will bind to NMDA, and then this allows magnesium 2+ that was previously blocking an ion channel to leave. And with a now open gate, calcium 2+ and other positively charged ions, or cations, can freely move intra and extracellularly, so like in and out of the cell, to help with like neurotransmission. And so the overexcitation of this receptor, which would mean more calcium moving in and out of the cell, is what's believed to promote neurodegeneration from like excess stress. But the exact mechanism is still a little elusive. And particularly in Parkinson's disease, this increased calcium traffic is associated with seizures. There we go. And so memantine works as an antagonist to NMDA. And so it'll come in and it'll take the spot where magnesium 2+ was. And even when the agonists glycine and glutamate are attached still, memantine will like stay in place and block calcium transfer from actually going, which then helps prevent this overexcitation of the NMDA receptor, and thus prevents more neurodegeneration and therefore slowing the progression of Alzheimer's. Cool. So what have we learned so far? We've looked at the two most popular hypotheses used to treat Alzheimer's, and the first was the cholinergic, where depletion of acetylcholine leads to less synapse function and eventual neurodegeneration. And this includes acetylcholinesterase inhibitors like donepezil, rivastigmine, and galantamine to stop the breakdown of acetylcholine into choline and acetate. And the second was how NMDA receptor overexcitation may lead to neurodegeneration, and how memantine is an antagonist to the NMDA receptor and blocks calcium from freely moving through that ion channel, which then will prevent overexcitation and therefore slows neurodegeneration as well. And even though these theories are what's used for Alzheimer's treatment, if you look at like the Alzheimer's Foundation website or Wikipedia or literally any well-respected informational Alzheimer's website, the most prevalent theory explaining neurodegeneration from Alzheimer's actually focuses on beta amyloid plaques and neurofibrillary tangles. So neurofibrillary tangles, we'll start there. Microtubules are these like tiny little tubes, surprise, surprise, and they're inside cells, including neurons. So on the left of this image, you have a neuron here, and you got this like axon coming off of it. And then within that axon, there are just a bunch of microtubules. And microtubules help just like transfer molecules and proteins within a cell. And tau is a protein that is on like the side of these microtubules and helps hold them together so they don't like fall apart and disintegrate. But in an Alzheimer's disease brain, this tau can become hyperphosphorylated, which then means the tau will come off of the microtubule will start to disintegrate, and the hyperphosphorylated tau will like clump together and like create this huge tangle within particularly like axons of neurons, which then will interfere with synapses and therefore lead to neurodegeneration again. Now the other aspect of this theory, beta amyloid plaques are clusters of malformed beta amyloid proteins. So like somewhere along the way of formation, these beta amyloid proteins became shapes they weren't supposed to. They became malformed. And that causes these proteins to clump together outside of neurons, which will then interfere again with synapses and leading to neurodegeneration. I personally like to think of it like when I say interfering with synapses, I like to think of it as me trying to walk outside in Appleton currently, because there are so many river bugs and they just hit your face, get in your way. You have to slow your walk. You sometimes have to swat them out of the way. It's a good time. Yeah. And so if these are the most popular theories for Alzheimer's pathology, then why don't we see more treatments that focus on those areas directly? And to do that, we're going to look at how beta amyloid plaques come to be actually. So we start off with the amyloid precursor protein, which is on that top left there. And APP can either be cut by alpha secretase into APP alpha, or it can be cut by beta secretase into SAPP beta, and then SAPP beta will then be cleaved by gamma secretase into the beta amyloid protein. And interesting fun fact, the role of beta amyloid in the brain and elsewhere is kind of unknown, but malformed beta amyloid highly correlates with Alzheimer's and is like the one actual sign we know, like if a postmortem brain has amyloid beta plaques and they were showing a lot of symptoms of Alzheimer's, then they had Alzheimer's, and that's kind of like the diagnosis. And so in early onset Alzheimer's, there's oftentimes a mutation in this amyloid precursor protein that'll then like go in throughout the whole system and just like, a good phrase is garbage in, garbage out. If you put in a garbage protein to then be cleaved or like be reshaped into another protein later on, it's going to be garbage still, which then will cause like this beta amyloid protein to not be how it's supposed to and instead will clump together into plaques. And particularly when APP has like a genetic mutation, both SAPP alpha and beta are like also malformed and can also clump together, so that's really fun. And then PSNE1 and PSNE2, the other mutation for early onset, those are both two like genes that help code for subunits in the gamma secretase protein that helps the final cleavage to get to a beta amyloid protein. So when there's a mutation there, it'll cut it up in the incorrect spots, which means the final protein can't fold properly and therefore will clump up later on. Again, beta amyloid plaques. And so targeting this pathway is a little bit difficult because first, beta amyloid isn't just in the brain, it's kind of throughout the body. And therefore finding drugs to only target the brain beta amyloid already requires extra layers and extra like detail and attention to detail. And additionally, I found this interesting mouse study that found that induced beta amyloid plaques in the peripheral system could then cross the blood-brain barrier and get beta amyloid plaques into the brain and induce Alzheimer's symptoms, which like adds more complication because it means you have to be really like aware of how the body works again as a whole system instead of just like looking at the brain. And then, yeah, there we go. And then there was also an attempt made to create a gamma secretase inhibitor for early onset Alzheimer's, but there were severe side effects to that, and it was like promptly like dropped off the radar, and I couldn't find too much about it post that. Now, this is a large graphic. It's a little overwhelming, and it's from a great review on potential Alzheimer's drugs in clinical trials as of July 2020, so a little late right now, and I'm waiting for the next July paper so I can find out the next Alzheimer's drugs in clinical trials. So the outermost ring shows drugs in phase 1 of clinical trials, and the innermost is drugs in phase 3 of clinical trials. Phase 3 is like, if a drug passes phase 3 properly, they can be FDA approved. That's pretty cool. So it's kind of like the final step. And I want to use this graphic to illustrate some of the commonalities in Alzheimer's drug research. And I first want to reiterate that there is no known root cause for late onset Alzheimer's, and therefore most of the treatments aim at symptom reduction, including like most of the drugs on this graph or on this diagram as well. So first, we have a few pre-existing drugs. So a fair majority of these are already FDA approved drugs used for other ailments. For example, lithium is a drug used to prevent drastic mood alterations in bipolar disorder, and metformin, circled in red over here, is a drug used to control blood sugar levels. And again, both are pre-existing FDA-approved drugs that are now being put back through into the system to see if they have any interaction with like Alzheimer's symptoms. Additionally, there are drugs that will focus on the same pathways or mechanisms as current Alzheimer's drugs, so that NMDA or cholinergic hypothesis. For example, AXS-05 and ABP 786, now circled in blue, are both NMDA receptor antagonists like memantine. Another commonality is the prevalence of long clinical trials and subsequent discontinuation of research far into the Alzheimer's drug line. The first example I'm bringing up is solanezumab, which it's a drug that was in clinical trials starting 2013, but just this year, so like, that's over 5 years later, was discontinued when it failed to show statistically significant reduction of Alzheimer's symptoms against a placebo. CAD-106, I couldn't find too much or like what exactly happened, but the project was dropped by its company after results were not promising enough in phase 3, but there were also no news on like restarting the project or if they had any other like prominent Alzheimer's research that they were going to like redirect like this funding or this research to. And then donanemab is the final drug I wanted to bring up, and it's kind of on like shaky ground, which is where also a lot of other Alzheimer's drugs are. It's moved on to phase 3 trials at this point, and those are expected to finish in 2023. However, while some completed phase 2 trials showed a statistically significant reduction in Alzheimer's symptoms, others didn't show this reduction of symptom presence, and it's kind of like a 50-50 thing. So it's leaving donanemab on a potentially shaky ground for FDA approval. And as we've gone through this, it may or may not be evident, but most of these current Alzheimer's drugs are heavily based on pre-existing research, whether that's pre-existing drugs or pre-existing symptom reduction targets. And that's not necessarily a problem. That's good. We use knowledge to then inform further knowledge. But when so much of the research is being done into symptomatic reduction, which is what most of these are dealing with, where does that leave research on understanding Alzheimer's pathology to eventually find an actual cure for Alzheimer's? And so what, in my opinion, would help increase efficiency in the Alzheimer's drug and research pipeline so that a cure is more likely to be found? And the first and maybe most important aspect I want to focus on is increasing efficiency in the Alzheimer's, or that would increase efficiency in the Alzheimer's pipeline, is increasing reliable early detection of Alzheimer's disease. The most obvious way to do this would just be to increase research into Alzheimer's pathology in order to study how the disease evolves to try and catch it earlier and earlier every single time to maybe find a root cause. Although it's been years and it's still hard, but it doesn't mean we should give up at all. And then the next thing we could do though is to just increase the availability of current diagnosing techniques. Specifically, if we could increase accessibility of amyloid or tau PET scans, which will say be able to tell if there are elevated levels of amyloid or tau in a person's brain, therefore indicating potentially Alzheimer's, and also volumetric MRI, which can then tell how much has the brain degenerated, if it has at all. Also, as of the end of last year, the company Eli Lilly has been researching and looking on to how to produce rapid blood tests that can stand for increased tau concentrations. And their early trials have so far indicated an 89 to 98% accuracy in predicting whether someone has Alzheimer's or not from tau levels, which is really interesting. This technology still is a few years away from being super mainstream, but this kind of idea of rapid blood testing to test for some Alzheimer's related hallmark could be a major step in catching Alzheimer's quicker, especially if it's streamlined and made cheaper and more accessible. Another area to look into is through genetic testing. So there's already a correlation for early onset of Alzheimer's with mutations in the APP and PSENE1 and PSENE2. But are there other genes that could be indicative of higher risk of Alzheimer's or late onset of Alzheimer's? Some genes have already been proposed, but they're all just a lot of words that I didn't want to put into this presentation. And they're not super well researched. There's just a lot of hey, maybe this has something to do with it. But it's worth looking into, especially with how much more accessible genetic testing is becoming. Might be a worthwhile path. And then also, I personally believe that increased public education on Alzheimer's could also lead to earlier detection. Those jokes of, oh, you can't remember blank? Man, you must be getting old. Throw those phrases out. Drastic memory decline with age does not have to be the norm, and the sooner we recognize it as atypical and we can show that to other people, the faster family members and close friends can pick up on those hard to spot warning signs earlier, as opposed to later when Alzheimer's has already progressed for those 10, 15 years. And then the next interesting thing I wanted to bring up is how virtual screening of drugs could help streamline preclinical drug design. And what exactly do I mean by virtual screening? Well, let's say there's a computer program that has all the data for acetylcholinesterase input into it. After training the program's AI, the computer can look into creating drugs that fit into the active site of acetylcholinesterase, maybe even better than donepezil. And instead of creating a bunch of similar looking molecules in a lab, by spending weeks in there and just making it together and throwing things into flasks, which is a lot of how current drug design is happening, which is also very time and money intensive. one could use like a quick computer program to narrow down possible drug design ideas, as opposed to like, weeks in the lab. And that's not to say this like is perfect, even though it might be much quicker and efficient. The problem again goes back to garbage in, garbage out. If an AI is poorly taught, then the AI will only produce like bad drug designs that won't work. And there are other potential issues in regards to what aspects an AI might look for. For example, let's say the AI comes with a theoretically impressive NMDA receptor antagonist, but the program doesn't run any calculations to see if it has any like negative drug-drug interactions with like, I don't know, like literally like Aleve, to see if it causes severe symptoms when it comes into contact with it. And in that scenario, this drug may get made, go through maybe a few preclinical studies, and then it'll get dropped somewhere down the line within in the clinical trial pipeline because it had this negative drug-drug effect. However, like if these pitfalls are kept in mind and accounted for, and especially with like an increase in like technology and understanding how AI works, virtual screening of preclinical drugs may become a vital step for Alzheimer's disease in the Alzheimer's disease treatment pipeline. I think it's already like getting there. It just needs a little bit more time. Now I want to talk about Alzheimer's disease pathologies that I believe could benefit from like more research, in addition to the already popular cholinergic and NMDA receptor hypotheses. And how I think like looking into these hypotheses in particular may reveal more information on Alzheimer's pathology itself, as well as provide more therapeutic targets to treat Alzheimer's symptoms in the meantime of us trying to figure out the actual root cause. And the first one is viral involvement. So the herpes simplex virus is present in about like 90% of adults, which like no one knows, and I think it's kind of funny. And it's what causes canker sores and like as such throughout like on your mouth. And normally this virus is dormant, but there's a correlation between Alzheimer's patients and a reactivation of the virus like later on in life. Whether this reaction is caused by Alzheimer's or not is kind of like up for a debate. But if there is a relation, it would be worth looking down into how the herpes simplex virus 1 inhibitors could maybe potentially decrease Alzheimer's symptoms or vice versa, something like that. Another path to look down is about dental health. So Porphyromonas gingivalis is a bacterium that causes a form of gum disease, and it's been found in higher concentrations in Alzheimer's patients' brains compared to non-Alzheimer's brains. And there's a positive correlation between gingipan toxins from that bacterium and tau protein levels. And currently, what's pretty cool is there's one drug, aduzaginstat, yes, that's in clinical trials. I think it's in like phase 2 at this point, to see whether inactivation of those gingipan toxins can reduce Alzheimer's symptoms. But those trials still have like years to go before they'll be like relevant in a conversation or like in an everyday conversation. Another thing to look down would be inorganic molecules. For example, zinc 2+ is an inorganic molecule that's been shown to regulate glutamate production. And glutamate is one of those agonists for NMDA receptor. Once it binds to NMDA, magnesium 2 can leave the calcium channel, and then like overexcitation could happen in Alzheimer's. So controlling zinc 2 levels in like a very directed specific way might be another way to then control NMDA overexcitation. Another aspect is the cardiovascular system is another like potential pathology interaction to look into. So it's known that hypertension increases peripheral beta amyloid proteins, and as we've seen in those mouse studies, it might be possible for that peripheral beta amyloid to then move across the blood-brain barrier and induce like Alzheimer's symptoms if it is malformed. In addition, patients with type 2 diabetes have a 65% chance increased risk for developing late onset Alzheimer's. And I briefly brought up metformin, a current hypertension reduction drug, or like a blood sugar controlling drug in phase 3 of clinical trials to see if there's any effect on that on Alzheimer's pathology. So like this little hypothesis already has some attention, but more research on the matter like never hurts. And then the other thing is a person's microbiome might also have an effect on Alzheimer's, because if you didn't already know this, your body is more like, you're more made of bacteria than you are of you, and the composition of those bacteria have massive sway on a person's health. So back to Alzheimer's though, on one study found that there was a distinct microbiome composition in 85% of dementia patients they looked at. So like very, very late stage Alzheimer's that is like complete cognitive loss. Additionally, peripheral Inflammation in patients with cognitive impairments was associated with increased of the bacteria Escherichia and Shigella, and those promote inflammation, and further associated with decreased U-bacterium that promotes anti-inflammation. And in Alzheimer's, persistent inflammation can increase stress on neurons, potentially accelerating neurodegeneration. It's one of those like smaller risk factors of late onset. The reason I put those two aspects on the same side is because both the cardiovascular system and microbiome are like partially, if not mostly, controlled by diet. Now, like, I love myself greasy foods. They're fantastic. Like burgers, fries, more fries. Those high simple carbohydrates, high trans fats, high cholesterol foods are lovely, but they're kind of like awful and terrible for the body if that's all someone eats. And the Western diet in particular is like very harsh on a person's cardiovascular and microbiome health. I mean, we see so many ads for like, wow, get your cholesterol down. And it's because like our Western diet is so tasty, but not the greatest for an everyday choice. Alternatively, so like that's bad. Alternatively, the Mediterranean diet has been suggested as a much healthier alternative, rich with vital like proteins and molecules that are deficient in a Western diet that may help slow any neurodegeneration waiting to happen in the brain. And this includes choline, because I mentioned it much earlier in this presentation. How choline like isn't really produced in the body; it's only recycled from the cholinergic system, or you have to get it in a diet, and it's very lacking in a Western diet. However, it is in a Mediterranean diet, which is pretty interesting. And to be honest, the most tempting thing about the diet, though, is that like hummus is included in that, and I'm like super food motivated, and like hummus is just amazing, especially with vegetables. And this is the last thing I'm talking about, I promise. And I wanted to end it on a more like kind of interesting, hopeful note. So I'm going back to the big graphic, and I want to point out one of these drugs, aducanumab, in a phase 3 of trials. And so it's pretty cool. New drug, question mark. And its story goes kind of like this. So, it also binds to beta amyloid plaques to like stop them, or not the plaques, malformed beta amyloid protein, so it stops them from clumping up to form those plaques. And so in 2019, its phase 3 clinical trial was halted due to an independent group coming in and saying like, hey, we're not sure if this drug is actually going to produce a statistically significant reduction of Alzheimer's symptoms. But then after that, it got some FDA help in designing new trials to study the efficacy of aducanumab. And so far, it's promising, at least more promising than some of the other Alzheimer's drugs in the trials that we've seen. However, the concern is that a super high dose is needed for efficacy compared to the current drugs on the market. And then May 5th, an FDA advisory committee met to approve aducanumab after these trials, and there was 1 yes, 8 no's, and 2 uncertains. However, Biogen and Eisai, which are the companies that are producing aducanumab, received an extension on their trials to see if they can like expand them and like get them even better. And they have 1 more shot to convince the FDA to approve aducanumab as an Alzheimer's symptomatic treatment. And that final decision is going to be made June 7th. I mean, unless they get another extension. But who knows? Maybe it'll pass. But it's definitely something to keep an eye out, because this is like the closest it's been in like a few years. I mean, like, I'm going to be looking up the outcome at least. Yes. So I know we covered like an overwhelming amount of information, and it was a long presentation. Let's return to that original question, which was, why haven't there been new Alzheimer's drugs approved by the FDA since 2003? And like the current therapeutics in circulation are effective at slowing down the progression of Alzheimer's symptoms. Because of this, a lot of potential Alzheimer's drugs are then heavily influenced by those mechanisms and insights of those 5 currently FDA approved Alzheimer's therapeutics. That is, like, potential drugs are not FDA, and if though, sorry, stumbling over the words, last slide. That is, if those potential drugs are not already FDA approved drugs to treat other ailments, or discontinued for some other reason. And there are ample interactions between systems of like Alzheimer's pathology as well as like others that are prime targets for like future Alzheimer's pathology and symptom reduction research. Again, my favorite being the cardiovascular / microbiome / Alzheimer's interactions, because like I'm just very food motivated. However, there's like forever more to discover about the world, and like every day I'm reminded of just how much information like we don't know about the body, and how like it has been since 2003 since we've like made major strides in Alzheimer's. And at some point, I'm sure we're going to find a cure, but the only question is like how long. But I have hope at least. Cool. Those are my citations. Thanks for listening. Please let me know if you have any questions.
Yay, very nice, very nice. Okay, lots of stuff. So who wants to start the questioning off?
Vivian, go ahead.
All right.
Sweet. So you mentioned like the beta amyloid proteins. Like they were, when they are made wrong, they kind of like clump up. Do you know what they normally do?
Yeah, so like,
Sorry, kind of random.
no, you're good. What they normally do is not actually known super well, like how they affect the body. There's not like a clear thing, which I find kind of wild. We just know that like when it does plaque up and when it like interferes with the synapse, it like leads to that neurodegeneration, which is why it's been just like claimed as this hallmark. And I think also, so like the first person to like coin Alzheimer's, like the guy who like found it and diagnosed it, first found these like amyloid beta plaques in a post-mortem brain, and that was kind of how Alzheimer's got its like distinct little category as a disease. So I think that's also why.
Was that person Alzheimer?
Yeah.
Yeah, okay.
Alois Alzheimer.
Yeah.
And so that's kind of like why I think that theory in particular sticks super strong regardless. Yeah, so who knows what amyloid beta actually does.
Cool.
Yes, Micah.
Yeah. Do you know if that high choline Mediterranean diet might help someone who has early onset Alzheimer's?
I'm not entirely sure of that, just because early onset Alzheimer's seems to be like controlled by genetic mutations as opposed to like other risk factors. So I think the way to like counteract that like 1% of early onset Alzheimer's would be to look into what those genetic mutations are and how to affect those. Like if there's some like epigenetics that's involved with that, I don't know about it. So at least for the early onset, I don't think the Mediterranean diet is a super viable option, but it could help like reduce risk of other Alzheimer's symptoms.
Okay, thank you.
Okay, well, let's go Lindsay and then Vince.
So you talked about the genetic mutations a little bit, but that like we don't know the exact cause of Alzheimer's. Is it that like the genetic mutations aren't like fully understood what they do, or do they like not account for all of the symptoms with Alzheimer's?
I'd say the better way to put it is it doesn't seem like those genetic mutations account for all Alzheimer's disease diagnosis. So like if someone has like that APP genetic mutation, it does not like, or some people may not have the APP genetic mutation, but they still will later on be diagnosed with Alzheimer's. So it's like just because it doesn't encompass all of them, it's not like its own distinct thing. Especially with like late onset Alzheimer's, which doesn't really have that like those specific genetic mutations that correlate with Alzheimer's, or at least that are known currently.
I was going to say, I think maybe you covered this in your talk, but would you say that we even know for sure that the beta amyloid plaque formation is causative and not just correlative with Alzheimer's?
Honestly, I don't like personally think so, but it is like one of the few things that if there's amyloid beta plaques, you're like pretty likely to have Alzheimer's, vice versa. Like it's a very high correlation that like is pretty like it lasts pretty well on like most Alzheimer's, but I don't, it's like not like there hasn't been like mouse models that induced amyloid plaque and then all of them got Alzheimer's or something.
Because I took a statistics class and now I know that causation is not correlation. Sorry, this is a little joke. Vince, want to jump in?
Yeah, so you had this massive graphic with like all of the different drugs, and there was a group, I think they were like epigenetic drugs. Do you know what they do? If not, that's absolutely fine.
Yeah, I'm gonna have to disappoint you here and say I don't know what all of those epigenetic drugs do, but I'm assuming they're focusing on the ApoE4 allele. So in late onset Alzheimer's, you can get like a few ApoE variations. There's like 2, 3, or 4 are like the most common, and with the presence of ApoE4, there's like I think it's like a 30 to 50, I don't remember the exact percentage, but there's like a 30 to 50 like percent increased risk of getting Alzheimer's versus if you have ApoE2, there's like a 10% risk or something. So I think those are probably going to be focusing on that or early onset Alzheimer's to see if it can do something to interact with like the APP or like PSNE1 or 2 genes.
Great. So I'll wrap this up with a statement and a question. For those of you that are interested in learning more about this, my favorite medicinal chemistry blog is called In the Pipeline. It's by somebody named Derek Lowe, who's a practicing medicinal chemist and goes into some of both the science and the political intrigue behind stuff like aducanumab and the battle that's gone back and forth. So really great resource there.